My name is Sandra Barnard. I'm a professor in botany and I'm currently the head of the Department of Botany. And we are going to share with you our research interests. Each person that lectures in the Department of Botany has his own research interest and therefore his own research field.
We are a team of 4 researchers studying different groups of freshwater algae in rivers and dams throughout South Africa. Algae range from small unicellular organisms to large filamentous forms that can easily be seen with the naked eye. Some algae are problematic, like those secreting toxins, those forming scums on the surface of the water, or those that cause problems in water purification plants. However, the majority of algae are beneficial, forming the base of the food chain in aquatic environments. And without them, life in water would not be possible. All organisms higher in the food chain, such as small invertebrate animals, snails, fish and birds, depend on them as food. Professor Net Jansen van Vieren and Dr. Anatoly Levinets study different types of algal groups in freshwater environments. These include the so-called cyanobacteria, a problem group that resembles algae, but they're not true algae. Some species can also produce toxic compounds that can be lethal to various animals or humans that consume the water. Most cyanobacteria are non-motile and can perform limited gliding movements. However, many members of other algae groups can swim actively throughout the water by means of hair-like appendages called flagella. Some can also move around by changing their shape, resulting in fascinating and spectacular movements. They are also used to monitor water quality by studying certain chemicals in the water that influence algae. The presence of cyanobacteria can indicate water pollution, while other algae can be indicative of clean waters. One algae group regarded to be excellent indicators of water quality is a group called the diatoms, on which my research focuses. Diatoms are a unique group of the algae with cell walls composed of almost pure silica. Diatoms are found floating in the water column or attached to rocks, plants and other hard surfaces in dams and rivers. These microscopic and beautiful algae are used to indicate environmental changes and pollution. Some diatom species are associated with high water quality and others with polluted conditions. Together with her fellow researchers, Prof. Sandra Barnard concentrates on harmful algae and their presence in our freshwater resources. She studies their special powers by investigating different factors, making them more notable compared to the other algae. This will include the influence of nutrient concentrations due to the different types of land use on their growth and the different environmental conditions leading to toxin production. Water will be the future limiting resource in South Africa, and the quality of water will become increasingly important as our water is becoming more limited. It's therefore important to know the threats to our scarce water resources.
Molecular biology is the part of biology that investigates what happens inside a cell, as well as the characteristics of large molecules such as DNA present in the cell. In particular, I use DNA sequences to determine the presence of different organisms in an area or an environment, and to determine the presence of certain toxin-producing genes in organisms. Molecular biology can be viewed as a specialized research field on its own, such as the developing of new techniques or the discovering of new functions of genes, or it can be viewed as a specialized tool that can be applied in various research fields to identify specific organisms or molecules. Currently, I work with several research groups on various projects in which I am responsible for the molecular or the DNA part of the research project, as well as the data analysis thereof through what we call bioinformatics or biological computer science. As such, I work with land and water ecologists to determine various aspects of interactions between plants and the environment. As a molecular biologist, I currently form part of 3 main research projects. In the first one, I isolate DNA from dung collected from various animals that live in the African savannas, specifically in the Kruger National Park and Timbavati National Park, as well as from the domestic animals in the areas surrounding these parks. These animals include elephants, zebras, buffalo, impalas, and cattle. In a similar project, I isolate the DNA present in soil samples collected from the Namib Desert to identify the different lichens that survive in these soil crusts. This will help us to understand their role in this ecosystem and therefore how we can manage and conserve this ecosystem.
Plant physiology is considered an essential ingredient for improving crop productivity, a continuing necessity for today's ever-increasing world population. The research done by the Plant Physiology Research Group at the Noordwes-Universiteit aims to provide an understanding of the physiological basis of various plant processes and the underlying mechanisms in response to fluctuating environments, which is of great importance for sustainable crop production. Furthermore, the study of photosynthesis is at the heart of investigating the effects of stress on plants and monitoring plant growth and development. Numerous multidisciplinary studies in the areas of agriculture, physiology, biotechnology, molecular biology, genomics, modeling, etc. have encouraged the interest in the in-depth study of the photosynthetic machinery as a tool to increase the growth and production of plants under different environmental conditions. Among these effects, drought will have profound impact on crop productivity in the future. A very critical role is envisaged here for systems biology in identifying metabolic alterations and stress signaling pathways, metabolites, and the genes controlling these tolerance responses to drought stress, and in engineering and breeding more efficient and better adapted new crop cultivars for dry areas of the world. In addition to studying the effects of environmental stress on crops, the Plant Physiology Research Group also explores the physiological mechanisms of weeds associated with herbicide resistance. Agriculture practices utilize herbicides to help manage weed populations. However, the continued reliance on herbicides as the principal means of weed control results in a selection of herbicide-resistant weeds. An unfortunate but predictable outcome is that herbicide-resistant weeds have become more common worldwide. The widely successful use of synthetic herbicides over the past 70 years has imposed strong and widespread selection pressure, leading to the evolution of herbicide resistance in hundreds of weed species. To conclude, environmental changes have a strong impact on plant production. This comprises the hampering of plant growth by extreme weather events, the occurrence of novel diseases, and increasing production due to elevated concentrations of carbon dioxide. Our research therefore aims to unravel the molecular and biochemical mechanisms underpinning the successful acclimation of crops and plants to a changing environment. For this reason, researchers of the Plant Physiology Research Group focus on the fundamental analysis of stress physiology and photosynthesis research.
The Northwest University's Botanical Gardens falls under the subject group Botany within the School of Biological Sciences. The history of the garden dates back to the early 1960s, when the botany lecturers at the time needed fresh plant material for the botany practical lectures. Today, I want to show you and tell you how our botanical garden contributes to basic science for sustainable development in our community. The botanical garden contributes knowledge and plant material in 4 key areas, namely teaching and learning, conservation, research support, and community engagement. Let's look at the first key area, teaching and learning. Students in botany cannot be trained in plant taxonomy without fresh plant material. To give the students exposure to the live plant material, we grow various plant species in the garden that the students use during practical sessions for taxonomy, anatomy, and morphology. The students also have the opportunity to collect plant material in the garden for herbarium voucher specimens that they need. All plant species and certain birds, mammals, frogs, and fish species that occur in the garden are identified with a name board. The name boards give basic scientific information on the species and the traditional or medicinal uses of the plant. The second key area is conservation. The Botanical Garden is involved in the conservation of rare and endangered species in our area. The garden takes part in plant translocation projects, and we also grow plants from seeds and cuttings to contribute to the conservation of species. The third key area is research support. In the garden, we grow a selection of different plant species: grasses, annuals, herbaceous, woody shrubs and trees. Some of the individual plant species are used by postgraduate students to study the medicinal properties of the species, while other pre-graduate students use the plant communities to study the birds or insects that occur around them. For each of the plant species in the garden, we have collected a herbarium voucher specimen for identification purposes. The data of each species is also entered into the Botanical Research and Herbarium Management System databases, from where researchers have access to the data of various species for their research needs. Community engagement is the last key. The Botanical Garden is not only open to students and staff from the Northwest University, but also to visitors from the public and schools. In the garden, visitors can learn more about the local grassland indigenous species that has adapted to the climatic conditions in our area. These species are excellent plants to be used for residential gardens. In the garden, non-botanical elements such as a geological display, sculptures, and locative literatures are used against the backdrops of plants to broaden the appeal across a wider audience and to enhance mission-based activities.
The herbarium was established in 1932 by Anthony Goossens, first professor and head of the Botany department, who was a prominent grass taxonomist. The current curator of this herbarium is Prof. Stefan Siebert. The herbarium holds over 30,000 specimens from central South Africa, which are used for educational and research purposes. It is widely recognized for its large, regionally representative collection of grasses of almost 4,000 specimens. In honor of the contribution of herbaria towards the role of basic science and sustainable development, I will be taking you through the process of plant curation in a nutshell. When collecting plant material, there are a few handy tools and equipment that can be used, such as a pair of secateurs, field labels, field collection forms, brown paper bags or Ziploc bags, and GPS tools. It is always best to press the collected specimen as soon as you can to avoid losing important parts, such as the leaves or flowers, which may fall off as time passes by. Plant press can be used as not only a pressing tool, but also a drying tool. For best results, you can insert the plant press into a drying oven set at 35 degrees Celsius. Once the specimen has dried out, it can be mounted onto a mounting board using wood glue. Weights can be placed on top of the specimen to ensure greater contact between specimen and mounting board. To identify your plant specimen, you can utilize the quick guides within your herbarium to match out plant features. Other useful tools for identification include microscopes to identify plant features and field guides and published taxonomic literature. The specimen label is generated using the BRAM software. The software is also a useful tool to catalog all these specimens within your herbarium. The last step of the plant curation process is to file your newly prepared specimen. After the label is fixed, the specimen can be transferred to a freezer for 48 hours. This is to kill any insects which may be present. The decontaminated specimen can then be incorporated into your main collection.
Forbs are neglected in the plant world. They represent plants other than grasses and trees, and for this reason they are not always seen as important. In fact, most people do not know just how important forbs really are. Forbs maintain vital ecosystem functions, including providing food to animals in African ecosystems, as well as food and medicine to humans. Researchers from the Forb Ecology Research Group, or FERG for short, study all the different important functions of forbs in African savanna and grassland ecosystems. FERG is a postgraduate research group led by Prof. Francis Siebert. FERG has strong collaborative research relationships both nationally and internationally. Our work at PHIRG is to provide as much information possible to the world out there on the importance of understanding ecology of forbs, to conservation, land management, food security, and to livestock production in African ecosystems. To fully understand the importance of forbs in grasslands and savannas, the research in PHIRG is subdivided into 3 main research themes. In ecology, the interactions of forbs with other plants, animals, insects, and microbes are studied, as well as the effects of disturbances such as fire, drought, and climate change on forbs, and how they can adapt themselves to such events. We further study the reproduction strategies of plants in different ecosystems to predict future communities, and finally also where forbs have invested for their future, for example in seed banks or bud banks. In medicinal and food plants, as a research focus, we study the medicinal and nutritional properties of forbs in Africa to make people understand that these plants are important to human livelihoods, especially for future generations. The third area of research includes molecular research. We isolate the DNA from plants from environmental samples such as herbivore feces to be able to analyze which species of forbs are preferred in the herbivore diet. With this, we are able to understand which forbs are important food items for both livestock and wild animals. These studies and others provide insights into the best practices for conservation, agricultural management, and for restoring sustainable ecosystems. Weeds, wildflowers, bulbs, and smaller shrubs, so the forbs, all photosynthesize just like grasses and trees. This means that they are also primary producers and are crucial to all life on Earth. Surely forbs then have important functions and resources to provide to animals, humans, and microbes, but they are also important to larger ecosystem processes since they capture carbon and hence contribute to cleaner air, they contribute to clean water in ecosystems, prevent erosion, and heal disturbed ecosystems. Therefore, we at PHIRG study forbs because they are extremely important for the environment and human well-being in the future.
Our research focused on land degradation and the impacts by different factors like climate, overutilization, improper management strategies, social and economic patterns, also called anthropogenic impacts, that lead to land degradation in arid and semi-arid savanna rangelands towards the western parts of South Africa. One of the main problems of land degradation in South African biome is the densification of woody species, so-called bush encroachment, due to some of the environmental and anthropogenic factors, depending on the type of land use and region. We mainly work in arid and semi-arid savanna areas in the Limpopo, Northwest and Northern Cape provinces, where bush encroachment occurs. The Department of Forestry, Fisheries and the Environment, especially the Division of Natural Resource Management, have a lot of projects where bush encroachment is controlled by local contractors with the help of the adjacent communities. After the bush is controlled, restoration or the improvement of the land has to take place, especially to increase the fodder production for the grazing animals by determining the biomass of the grasses. We also call it improve the grazing capacity. The main aim of our research is the monitoring and evaluation of the restoration and rehabilitation practices after bush control to improve soil conditions and increase the grazing capacity for the livestock and or game which is kept by the land user in communal, commercial or conservation areas. The research includes helping community members, especially females, from the formerly disadvantaged communities in the training and capacity building activities like data collection, monitoring, developing of Excel sheets and reporting. Why do we do this research and how do we contribute to sustainable development? The research group's objective is to restore or rehabilitate the integrity of degraded and bush encroached rangelands by addressing the loss of biodiversity, ecosystem functions and processes through ecosystem services using sustainable land management practices. The livelihoods of the persons that participate in the research activities is improved and the research contributes to poverty alleviation. Restoration projects are also carried out at international level and include the participation of neighboring countries in the SADC region.
Our research on cities, towns, villages and settlements include basic as well as applied aspects. The basic aspects focus on biodiversity as well as ecosystem services. Ecosystem services are basic ecosystem functions that benefit humans. Urban wetlands such as this one of 45 hectares in the middle of Potchefstroom does not look spectacular in winter, but it is multifunctional. It's not only a habitat and breeding ground for birds like thick-knees and marsh owls, and also the last place along the Moor River where a large population of one of the torch lily species can be found, but it also buffers extreme events such as floods. There are 2 large stormwater outlets in this wetland, and the wetland also purifies this polluted water. This is what this wetland looks like when the torch lilies are in flower. It's quite a spectacular sight. The concept of green infrastructure is used to describe the areas that we study. Green infrastructure is an interconnected network of urban open spaces. In our applied studies, we determine the perceptions of different stakeholders, including urban residents, on issues such as urban biodiversity and also ecosystem services. Okay, let me give you one example. We currently study health clinic gardens as a special type of community garden in the entire Northwest province. We want to determine the value of the gardens, even the economic value, and also the type of knowledge of the different stakeholders. Information is integrated into management plans to provide the knowledge about gardening and also the useful plants and how to increase food and nutrition security. Our applied studies also include planning and design of sustainable urban areas, and we promote the value of urban green infrastructure planning through important principles such as the increase of connectivity between different green areas, and also to integrate the built environment such as buildings, roads and parking areas with green infrastructure such as bioswales, green roofs, rain gardens and so forth. So why are we doing all this research? Because we live in an urbanizing world. Close to 60% of the human population lives in cities, and it is predicted that about 90% of the future urban growth will be in Africa, Asia and South America. Although urban areas only cover 3% of the land surface area, they account for up to 80% of energy consumption and at least 70% of carbon emissions. Increasing urbanization therefore destroys natural habitats, biodiversity and also ecological processes. And all of these lead to poor human living conditions. Detailed urban ecological studies that provide urban decision makers with information and tools to plan, design and manage sustainable and resilient cities are therefore necessary.